Rewritten human history. Archaics and hybrids are created and bred in cradles before they are migrated, carried on ships or otherwise, and involved in the population turnover events. One after the other, like a car factory spews out cars and models, that are followed up by peopling, populating, of significant parts of the world, and mixing. Hybrids and archaics can be recognized and identified by their haplogroups. At the end, or rather the actual groups and mixes we call modern humans. Most probably modern humans are also hybrids, and became so the same way as archaics did, but obviously modern humans are not archaics, simply because at this point they are not extinct. This history all started out in the Americas, with Y-DNA haplogroup Q and blood group O, because the ancestral markers clearly point this out. Y-DNA haplogroup Q and blood group O confirm each other in both directions, but there are still other markers that also confirm the same. Cradles are the start. Archaics were hybridized and possibly bred and interbred, on purpose and dedicated for a purpose, in cradles by contemporary humans. Please remember that 60% of the food we eat today was hybridized by humans in South and Central America. Most probably this also happened in cradles as well. Scientists today do not know how these humans achieved this feat in the past, and they still cannot replicate something similar until today. What our scientists achieve today is called by other scientists and themselves, the harvesting of low-hanging fruits. This way, pointing out that what we are able to do today does not come close to what they know happened in the deep past in the Americas and probably later in other continents. On purpose and dedicated. As said, these hybridizations are executed and on purpose, and also dedicated, this by non-archaic humans and probably always departing from quite original humans, DNA, this for DNA stability reasons, and not from archaics, which are genetically manipulated and consequently hybrids, DNA, not one archaic interbreeding with another archaic forming and interbred hybrid, which is fundamentally different from a genetically manipulated hybrid. Leading to instability. Any manipulation already leaves a less stable DNA, that consequently is less suitable as a basis for yet another manipulation. Too much manipulations could lead to something similar to founder effects. Genetically manipulated most probably is about disturbing DNA, for instance with radiation, and the DNA reorganizing itself, with or without humans providing it additional material. In other words, in our view no nuclei of cells are open to manipulate DNA, as it does happen today. This DNA reorganizing, that is inherent to DNA, is proven to happen outside bodies, even to the point of forming organs like independent brains. Without bodies, that are used in research, this at least to some point of completeness of the organ or brain.